So what you're looking at here is actually the hyphae and the spores of a beneficial surface fungus called Penicillium nalgiovense. This is actually the mold on the outside of a salami that I was curing. So, and this is a picture, the little round balls are the spores of the fungus, and you can even see the one in the center. You see the hyphae that's poking out, is growing out of it. So that's a germinating spore, and then the Y-shaped structure to the left is actually the intact hyphae of that Penicillium mold. Pretty cool. So this is a food microbiology, not soil. Here's more of it. This is, I believe, some fennel pickles that I made. So lactobacilli are going to be really long and square rods. These are some other bacteria floating around in there. There's lactobacillus, the longer ones, probably lactobacillus. See all the different bacteria in there? Look at all the different morphological shapes. That's, see the mouse. Yeah, these diplobacilli, right? Like two bacillus attached together. Those almost look like yeasts. Those ovular things, I'm not sure, but that's pretty out of focus, pretty out of focus. Okay, you see the long skinny guys? Those are lactobacillus. This looks like a ciliate. Hard to say because it's not in focus. Could be a cyst of some kind, but yeah, and yeah, the chunks, and then the long rod-shaped things seem to be like, there may be some like down here. Is that fungal? Kind of looks fungal, but like this guy and these guys look like lactobacillus to me. And the presence of bacilli, it's like, we may have some anaerobics stuff going on here. That looks like actinomycetes to me. Just anaerobic. I mean, I can't tell if this is soil or if this is food. I don't remember when this picture is from. So it could be a situation where I wanted it to be anaerobic because I was fermenting something, but that was the fennel pickles, I think. Crazy activity. And those things were shooting around. One of the things about Photo Booth on the Mac is that you can't take movies. With the ScopeImage software on a PC, you can take movies of the motion that you're seeing, and it gives you better clues as to what was happening. But those bacteria were just going wild in there. I think that's a sprouting spore, so I took a picture of it. We're moving on to compost photos now, for sure. There's some actinobacteria right here. But it's just one piece, it's no big deal. It's not going out of control. Here's cellulose, probably from the wood or some paper that was in the compost. There's a fungal spore. That was a ciliate. It's really hard when you're taking pictures of fast-moving things to get the cilia that would have been coming off the back of him, but he was zooming through there. There's a nematode. It's just the bulb on the bacterial nematode that I was taking a picture of. There's this mouth. There he is again. Oh, that looks like a dividing protozoa of some kind that I was excited about. I'm gonna take a picture of it. Looks like some maybe plant tissue, maybe, I don't know. I was thinking it was fungus, but now I'm looking at this part right here, and I don't know, but it certainly looks fungal to an extent. Sometimes I take pictures of things if I don't know and I want to go back to it. Like, what are these? Like, is this some kind of emerging amoebal cyst? Like, is this the pseudopod of the foot of the amoeba, and this is the test that it was in, and it's coming out for some food? Or is this a dividing an amoeba or a protozoa undergoing fission, which is how it reproduces? Like, I don't know. So I take the picture so I can go back to it later. There's a cyst, amoeba cyst. See the double wall there? There's something. Don't know. That's some kind of egg sac. I think I was looking at this guy. Is that an amoeba? What is that? I don't know. Fungus. Probably a scratch on my cover slip right there, or a water streak. You be careful about that stuff. That's the Mickey Mouse pollen. That was apparently like so exciting. It's some like conifer. I don't know what it is. Again, like something coming out of its cyst, which is cool. Some plant tissue. You can see a little bit of fungal hyphae coming out of that aggregate. Pretty thin, pretty clear. I don't know what I was taking pictures of here. There's an amoeba. It looks dead. I was taking pictures of these. I was finding these in the pasture soil a lot. I didn't know what they were, and my mentor said she thought that they were eggs. But I've looked at morphology of different parasite eggs and haven't found any that look like that, so I don't know. There's some fungus. That might be a big old cyst, like a big old amoeba cyst. Fungal spore here, aggregate. This I would call a cyst. This I would call a spore. Spores are the inactive fungus, cysts are the inactive protozoa. We got that right? Difference there? Here's a spore, probably. Maybe another spore. Some spores, that was like some plant tissue I thought was crazy looking, so I took a picture of it. There it is at 200x. So there was 400x. There's 200x. That was like, ooh, soil shrimp. So I think that's probably a piece of a microarthropod. There's that amoeba I showed you before that I said, where's the content? Where are the innards? I see a little bit of the plug here, but I don't, what's going on in the rest of the test? Maybe it's out of focus. I can't tell. What is that? Spores everywhere. Little fungal fragment maybe. There's that antenna I was telling you about. Isn't that cool? Like a little feather. So there's a fungal spore. See how it's segmented and dark like that? Probably pretty good guy, just how big it is. But some of the bad guys are pretty big too, actually. Could be an egg. Fungus, clearly. All these little round guys. You'd have to focus in and out to see. I don't think it's a air bubble. It looks to me like it's probably a cyst. What is this oblong thing? What's going on there? You know, you'd have to play with your diaphragm, look at how it's moving. Is it moving? There's another one of those ring structures. What in the world? Yeah, I couldn't tell if like what I'm seeing inside of it is just the rest of the soil solution. See what I mean? Or if it's like internal organs of a cell, in which case I would think of it as an egg, maybe like a parasite, a worm egg or something. But I can't find any with morphology that match. Here's another spore. Another ring guy. Weird. Strange things are happening. That's probably cellulose. There's a testing amoeba. They'll be like flat. They look like little skulls.
Pretty translucent.
Didn't know what that was. I think it's probably something that's turned, so you can't really get perspective on it. A little egg sac or something. There's some fungus. That's interesting. I don't know what that is. It could be a not so good fungus that's like, I don't know. Because I see some septae, but not a lot, and it's like a mass. It could just be like some tissue, but it could also be some bad guy. I took a picture further out. Didn't know what that was, but you see these little bulbs on the end. It almost looks like those fungal reproductive sites. So wasn't too pleased about that. I'm trying to get to that tea. This is all kind of the same compost, so it's not that exciting after, but there's some really good looking fungus. That was, we were thinking nematode eggs. To be sure, you'd have to focus in and out and see if you could see the little worm coiled up in there. And you never know if you're looking at a nematode egg whether it's going to birth a bad nematode or a good one, so you can't really be sure that it's exciting. But, I mean, I thought it was exciting just because I'm a geek. Like, that might be a... What is that?
A little fence around a odd shape.
I mean, well, the thing is like, without looking at the active slide, I don't know if these things are on top of it or inside of it or behind it, or like this could be a mineral, a soil mineral particle, and these could just be little spores that were floating past it. So looking at the pictures, it'll help you remember things, but it's not necessarily going to tell the story the way looking at the sample is going to. There's another fungus. Nice size. What is that? Some kind of egg, maybe. There's an amoeba. More fungus. Something broken. Some kind of mineral thing broken. Lots of aggregates there. Sometimes I take pictures just to look at that. Some type of mineral aggregate. Sometimes I take pictures just to remind myself, like if I'm taking a sample, I'll take what I call a representative photo. So a photo that looks like most of the slide looked, and then I'll just name it like greenhouse bed 2 representative photo or something, and that way I can remember sort of what was going on with that soil when I looked at it. More fungus. I take way too many pictures of fungus because I like it so much, but there's another testate amoeba. That was weird. I didn't know what that was. It was not moving though, so it's probably not concerning. Plant material. This was like vinegar spray trials, I think, that we were doing, and there's nothing that exciting going on here. Maybe some cyanobacteria. I don't know. It might be too thick. I didn't know what that was. I don't know if it was like part of a microarthropod or... Maybe. There's the tea I wanted to find. So these are my Lactobacilli. You see them? Long, square. So that's like, things are anaerobic-ish here. Not so good. You can see it's not very alive everywhere else. I mean, it's like there's not it's not teeming with bacteria. There's some bacteria here and there. There's some Lactobacillus, which tells me those are anaerobic bacteria. I've got some chains of rods there, which is just to show you different morphology. This is from another tea. This is tea from, I believe, 5th season that I tested for him last week. It was looking all right. I saw some fungus in there. There was some baby, there's a sprouting fungal spore. Pretty cool. That was in his tea. And then there's one picture, which you can barely see it, of a baby. There's a fungal spore. Oh, that's a flower petal. Trying to find it.
What's the shelf life on your tea, the tea that you would buy from 5th season or any other place before you would start seeing that?
Start seeing what?
What we're looking at.
Well, that tea was about 24 hours old, but it depends really on your brewer. My experience with looking at tea is like, maybe we have a huge brewer out here with a super powerful aerator, and I'll find sometimes if I look at it after 16 hours, it's had too much, It's run out of food, and it's got ciliates in it, and actinobacteria, and there's not a lot of life, whereas he's brewing its fifth season in these little 5-gallon buckets, right? And he's got different starter mix, right? So it depends on what's in your starter mix. But after 24 hours, his tea was looking pretty good. I have a feeling if I had looked at it a little earlier, it would have been better. So I think we're going to do some more looking, at his product. But, he gave me some of the stuff that he was putting in it. And it's, he's using some like some lobster compost and stuff like that. So he's got some really rich materials going in. This, you can't see it very well, but this is one of those little nematode larvae I was telling you about that I was just watching them go crazy throughout and eat all these little bacteria. And it was impossible to get a photograph of it, but that's the best I could do. But to see a fungal fragment like that in a tea that's been bubbling is pretty good. Because they can't stay intact very easily in a constantly moving system like that. That's it. That's all I got. Unless, here's a nematode in the pasture soil. I think that's at 10x magnification, so you can't see the mouth parts at that point, but it was a bacterial feeder. I'm trying to find examples of, there's actinomycetes after we sprayed vinegar. Here's a picture of a fungus that is being killed by, being killed by anaerobic bacteria. I don't know. You can just go, this stuff, you can go on and on and on, get lost in it. Do some Googling. Google insect larvae under the scope. Google, mycorrhizal. So just like any ecosystem, if you think about your soil as you would think about a forest, as you would think about an aquatic ecosystem, any disturbance keeps your soil and the compost that you're making in an early successive state. So this is what we're talking about when we see heavily bacterial soils and compost. They're going to be in an early successive state, and they're going to sort of favor a weedy, not very mature, not very dynamic system. So any management decisions which prioritize stability, maturity, and connectivity are the best. And so when we're talking about things you can go home and do right away, things that I've come away with this and started doing right away, always combining perennial and annual plants in my beds. So
Do you have a list of sectors?
I don't necessarily do it based on sometimes I'll look up what's a good companion for this or that. But mostly I'm looking for diversity in space, time, and architecture under and above ground, right? So I want things that are going to be maturing at different rates. I'm going to be looking for things that are different heights, different root systems. So I almost never anymore plant the same thing in one bed. So I'll do some beets, a block of beets, and then I'll do maybe a block of lettuces. Lettuce being a very shallow root, beet being a deeper root. Then I'll plant have in the middle just a random perennial valerian or something, or rhubarb, or just anything that I want to plant there that I can harvest the flowers from, or that is good medicine, or that's beautiful, that maybe it attracts pollinators. It doesn't necessarily have to have one specific purpose, but the purpose is that it anchors and provides a refuge for any mycorrhizal vesicles that can remain in the soil in that bed. So even if I'm pulling beets out here and onions there, I'm not decimating all the chances that mycorrhizae has, or that any stable soil organism, a soil organism that needs mature, stable conditions, that it has somewhere to go, or that there's an area of preservation, basically. Another thing is to just basically try as much as possible not to till. And so, there are people who till just the very top, very light dusting to get weed seeds, to knock out weed seeds, but they're not disturbing, obviously the most aerobic portion of your soil is going to be the top maybe 6 to 8 inches or so, so even tilling that deep is going to disturb a whole lot of life, but yeah, I'm working on like no-till to the best of my ability in my garden. And yeah, intercropping, like putting in between like your potatoes, like if I got a row of potatoes here and a row of potatoes here, I can put a row of leeks in between. When I hill the potatoes, I'm also blanching the leeks, like something like that. It's gonna give me a little bit more diversity. Also, like I always have in my non-growing spaces or walkways, I'm putting pretty unrefined wood chips or leaf mold or something like that. Crazy good leaf mold that Pat gave me that I looked at under my scope would be an awesome thing to be mulching a pathway with, especially if it's not highly refined, because even though you are going to be walking on it, if the particles are big, you're not going to be compacting it so much that it goes anaerobic, but it's going to be a really stable carbon source and a really rich food source for nematodes, for fungi, for stuff like that, you know? And it's going to be there even while things are changing in the growing spaces. The other thing, like feeding the soil, well, we'll get to that. Knowing that what happened before still affects you now. So this is what we're talking about when we say, oh, there was fungus here at one time, it all died, but we still have its awesome carbon bodies like giving us structure. So that's a great thing. If you look at a soil that's really, really red, it shows you that at one point in time, it was super anaerobic and that iron was oxidized in that soil. And even if it's aerobic now, there are markers left of its anaerobic past. You can also look at the life in the soil. Sometimes if you see a few ciliates, if you see a few lactobacilli, it could be that it's not going anaerobic, but it's actually coming out of being anaerobic. So look at your other context clues and do time course samples. One of the things about lactobacillus, those long, skinny, squarish rod bacteria, is that they will fight off other anaerobic bacteria. So they're not the worst kind of anaerobic bacteria to have in your soil. They're just a cue. But, you know, if you're seeing them and actinomycetes and ciliates, you know, and low aggregation, you might think, I don't have very good aeration. And just thinking about like, okay, if I'm putting fungus in the soil, you know, even if I'm renting my house and I know I'm moving out in a year, if you can tend that soil in that yard and fill it full of fungus, then you're doing everybody a favor. You know, even if the next person comes along and kills it, you know what I'm saying? You're still leaving carbon in the soil. So that's really powerful for me to know. The other thing is like feeding my soil really good compost and really good compost tea instead of concentrated fertilizer. So this is like, I want something, if I want dynamic life in my soil, then I have to provide food for all those trophic levels. So I need complex food that's not too concentrated. If I start putting over and over again a lot of concentrated and over-refined mineral amendments in my soil, then I'm going to end up with higher salts. I'm going to end up with funny exchange of nutrients in the soil, and I'm not going to be like, all the organisms are going to be like, well, this is no fun. You know, we don't have work to do. So it's no longer an option for me to be like, oh, I think I'll start a compost pile, or I won't. It's like, no, I got to have really good compost. And I got to be getting it looked at by somebody or looking at it myself. And if it needs to be improved, I got to figure out how to improve it, you know, because that's the primo way for feeding my garden or my plants. I've learned that any way that I can oxygenate or aerate problem soils is like a sure first step to ameliorating the problem. And also just that community offers context. So if I see one thing that's concerning, I'm definitely not going to freak out. I'm going to be looking at the entire picture to get a sense of what's going on, and that you're just learning and learning. You never know all that you can know.
Back to the oxygen and aeration, what formulas are you using?
Formulas? Like what would I do to aerate? Well, even just like putting a broad fork in your soil and just making air spaces in it. You know, if you saw that it was... So this is something I think about with pasture soils and something that I'm interested in looking at. So if I'm constantly trampling that soil with hooves and animals and putting heavily bacterial manure down on that soil over and over again, what's going to happen over time to the profile of organisms in that soil? You know, depending on the way I impact it, I'm probably going to see increased diversity, both in the plant species and in the microorganisms. But if I'm overgrazing, if I'm over trampling, If I'm coming back too soon, I'm going to overload with parasites. I'm going to have heavily bacterial conditions. It also affects other management decisions. Like we were thinking about using the vinegar to spray under the fence line to kill. So instead of mowing and weed eating around all the grass that grows on the fence line, I thought we'll spray the wood vinegar. And I said, well, hold on a minute. If you're mowing, you might be using fossil fuels and you might be doing more work, but you're not necessarily doing something to the soil. If you're spraying with a vinegar, sure, you're going to kill the plant, but you're going to knock out a pasture mix, kill it, and make way for an invasive species, first of all. And second of all, you're probably going to be putting that soil into an anaerobic situation under the fence line. And unless you hit it with aerated compost or aerobic soil afterwards, then you're probably going to create a situation in which those anaerobes can leak out into the pasture. Okay, what happens in a pasture system if you're spraying under your fence line with any organic acidic pesticide or herbicide, and then your animals are coming and dumping manure and trampling all over? Like, is that the best management decision, to be creating an anaerobic condition over and over and over again right at that fence line? You know what I mean? It may be fine if for example, at my house, I have some Norwegian spruce trees, big, beautiful trees, and they're completely overtaken with English ivy. So we've girdled all the English ivy that's up on the trees, and now we're thinking, what can we do with the stuff that's going on the ground? Well, in that situation, I say, why not hit it with full force wood vinegar, kill everything, and then hit it with some super aerated compost, like some really beautiful conditions after you spray. Because what happens after you spray is what's going to determine whether the bad guys win or not, So just like knowing what thinking through, like, well, in this case, it may be a great management decision, but in this case, it may not be a great management decision, or it may just depend on what I do next.
What's the acidity of that vinegar, or what you're using from the?
I couldn't tell you exactly, and we're testing it at all different dilutions. So I don't necessarily formulate the sample, I just look at it.
It's some agricultural vinegar for broadleaf control, and I'm not a big fan of spraying anything, but it was considered organic. So.
Yeah, and I think it's great. Like, one place I want to use it in our pasture is for spot killing thistle, like right now, this time of year, or like spot treating problem weeds, which are usually an indication of some kind of mineral deficiency. So they're telling you something. You can't just kill the weed and be like, oh, I have no problem. You got to figure out what that problem is. But you also do have to make sure they're not going to seed and, spreading. So I think the vinegar is going to be really useful for that. And then at different dilutions, a lot of research is showing that the vinegar actually acts as a biostimulant. So if it's not full force and the pH isn't super low, is it actually stimulating the soil life? And so that's something that we're also looking at. There's infinite variations, right? So.
Is that just white vinegar, or what kind of vinegar?
Oh, we're talking about the wood vinegar that comes from pyrolyzing biochar. So it's like a byproduct of making biochar. But you can buy horticultural vinegar, or you can mix distilled vinegar.
The type that you typically have in your house is a 5%. The other type that you would buy for agriculture.
It's higher, 30, yeah. But I've seen like old school poison ivy spray recipes that are like some kind of mineral oil with regular household vinegar. And there's supposedly in some soap, and it like works really good. Because you got to have like an acidifier and a surfactant and then like something else. So they're formulated. Yeah. So do we have questions? Head scratchers? Things you want to know more about?
The aeration of your soils and such, you can do that a lot. Question there with cover crops. If you can work that into your crop rotation with daikon radishes and such as that.
Yep, daikon radishes. Also in the summer, sudex. If you plant it and let it grow and then mow it, it'll shove its roots down further into the soil. Clovers are really good for aerating as well.
That works well with your multiple stories of planting and cover. Of your white clover's low when you get your massive red clover, mammoth clover.
Cover crops are so good for the soul too. Just like watching them grow there and having them there. Yeah, cover crops are definitely the best way, but like, this is why some people say, okay, well, chisel plow once or chisel plow every 5 years or something, because if you are in a management situation where you're worried about compaction or loss of oxygen or something over time, then you may make management decisions that call for a little bit of cultivation, to get the air back in there. It just depends. If it's a compost, maybe it needs to be turned. Maybe it needs to be rebuilt. Maybe, you can take material that's gone south and recompost it with fresh material and see if you can't revitalize it, But one thing that farming has taught me is that almost anything can be healed, You just have to know the right formula and you have to work at it.
So about the compost, let it sit. Don't be turning it constantly.
Right.
But then you're putting it on the ground, on your beds or whatever. Now it's
Like you've disturbed it in order to distribute it. Yeah. Well, I mean, this is why people who are really into, like the guy that I was telling you about, the Johnson guy, like this is why he's making like extracts with it and stuff, or he's coating seeds with it rather than like trying to disperse it in a spreader or like put it all over the soil, because it stays more intact or it's disturbed less in that way. My sense is that like if the life is there and you get it, and if you get it on your soil, it's going to find a way. I mean, if you literally have that much fungus, fungi in your soil, I don't care if you stick a shovel in it 500 times, put it on your garden, some of it's probably gonna take. And then there are people who say, like, if a compost is sat for a certain amount of time, then you need to feed it, right? So you would give it like oat bran or oatmeal or like things that the fungi want, Elaine says no molasses, it's too simple. It's too much of a simple sugar. It's a bacterial food. So like fish hydrolysate, oat bran, steel cut oats.
Complex carbohydrates.
Yeah.
What if you was going to make leaf mold and you wanted to accelerate that process and you use the fish hydrolysate, which is basically fish emulsion?
Isn't there a difference between emulsion and hydrolysate, the fish products? I can't remember what it is either, but there is a difference. It's important to note.
Okay, well.
One's hydrophobic, one's not.
Okay. The thing about the hydrolysate that I found is that, especially in compost tea, I mean, not in compost, is that it makes a lot of bioslime. It's like kind of gross when you're trying to like filter it and get it ready to go.
Would that be because of the nitrogen?
I guess. I don't know. Probably. But you're making leaf mold, so maybe it's not an issue for you.
Yeah, I found, I made a bunch of potting mix. I made about 2 cubic yards of leaf mold that I started back in 2016. And use that as my main mixture in the potting soil.
Oh, nice.
The compost that I had to buy from a local composter. But everything works and it's working really well. You can grow 8-inch tomatoes in there.
Yeah, I do that.
You should have brought some leaf mold for us to look at. Pond scum would be great to look at too. I took a sample from one of our ponds here where we're collecting rainwater, and I was so excited. I was like, oh, there's going to be all kinds of stuff in there. It was safe as safe can be. There was nothing in there. I was so disappointed. Well, I was like, y'all go swimming. It's fine.
Meredith, do you know if there's a difference between ramial wood chips and just like tree wood chips in terms of developing your fungi?
Oh, like the quality of them for the fungal food?
Depends on what kind of fungus you want to grow, or specific with turkey. Because certain fungus are, if you have a good mix, you're going to attract primary and secondary and tertiary composers, which is really what you want to do. So you don't have, I found just great to just use it.
To have it there, yeah.
But if you're targeting a specific mushroom, you have to know what their primary source is. And it's going to be either lignin or cellulose. And the lignin is on the outside of the tree, and cellulose is, well, it's all in it, but it's mostly concentrated in the center.
But different kinds of trees have different ratios of lignin to hemicellulose and cellulose. And so, I mean, ramial are mostly from hardwood species, right? Am I right?
You got to think about if there's any allelopathic, I have to say that word.
Yeah, allelopathic, yeah.
In other words, like black walnut, like
Is there anything besides black walnut that needs to be avoided?
Certain things are.
I don't know.
The chemicals that great.
Hardwoods, just stick to hardwoods.
Yeah.
I'm not picky myself, but I know like Michael Phillips says that he uses specific wood chips for his fruit trees for the mycorrhizal associations.
That's what I'm saying. You gotta figure out which, you're really trying to target something.
Do you know a good source for that? Like for that and for figuring that out?
I would talk to, you might want to, down west they're doing a lot of this stuff. And you ever read that book, Radical Mycology?
No.
Well, there's a guy out there, Peter McCoy, who's all into this low-tech mushroom farming and cultivating.
Tran Connor does a little bit of work with that.
Peter's into like, yeah, either one.
I mean, I don't really have both options at this point. I mean, ramiol's harder to get.
Yeah.
He'll
Sure, it's
either,
the outer limb
if he
trimming.
doesn't know, he'll tell you, he'll know who.
Uh-huh.
Yeah. And he's really responsive. Peter McCoy's really responsive.
Okay.
That's cool. I'm gonna take that, put it in my pocket.
That book is amazing.
What's the book? I've
Called
heard of
Radical
it.
Mycology.
Okay.
It's really awesome.
You came to Asheville.
Actually, I have a copy of it.
Did he? Did you go? No, they go on tour too.
Yeah, they do. They're having one right now.
Oh.
From Oregon.
Radical mycology, that one.
I like it. All right, other thoughts, other questions? We should be expecting some food pretty soon. I hope the class has given you some go home and do it now types of things to increase the good, higher taxonomic levels of organisms in your soil. And if anybody needs samples tested or looked at, feel free to give us a call at Living Web because we definitely want to provide that service. Obviously, there's only so much we can do, but I'd like to do more of it. So if you ever are curious about your leaf mold, holler. Yeah, thanks.